gives me great pleasure tonight to introduce our speaker, Dr. Terry Gasland. And she is a professor of computational genomics in the Marine Biology Research Division. And she leads the exciting new Scripps Genome Center here at Scripps Institution of Oceanography. Now, the center focuses on marine genomics, comparative genomics, and environmental genomics. And it will address important ocean issues, as well as issues related to human health and the environment and other areas. I just want to tell you a little bit about Terry. In 1984, she earned a bachelor's in computer science and Russian. I'm very impressed by that. From Duke University, where she was an Angie B. Duke Scholar. And she continued her studies in computer science at the University of Maryland, where she received a master's in 1988 and her PhD in 1992. And prior to joining SIO in 2003, Terry was Associate Professor of Computational Biology at the University of Rockefeller, where she established the Lab of Computational Genomics in September 1998. Now, she's a founding officer of the International Society for Computational Biology, and she was named a Sinn-Scheismer Scholar in 1999 and a Burroughs Wellcome Young Investigator in Molecular Parasitology in July 2000. And in October 2000, she received a Presidential Early Career Award for Scientists and Engineers. And many of you will know that that is the US government's most prestigious honor for young researchers. And in 2002, Crane's New York Business Magazine named her one of New York City's 40 under 40 rising stars in business, science, and the arts. So you can see we're really very lucky to have Terry come and join us here at SIO. So she's a computer scientist who has a long-standing interest in medical and biological questions. So Terry Gasland really designs tools for interpreting the huge flood of data that's being generated by genome sequencing efforts. And she has focused a lot on microbial genomes from very small organisms. And so her work has very important implications for understanding infectious diseases and pathogens in general. And recently she's focused a lot on comparing different genomes and also the gene expression data from multiple different kinds of organisms. And this is really pioneering new methods and new applications in comparative genomics. And so it's a great pleasure this evening to welcome Dr. Terry Gasland, who's going to talk to us about marine genomes, windows into ocean life.
All right, well, thank you for such a wonderful introduction. So it's really exciting to be here. Actually, coming to Scripps for me kind of brings together many different aspects of interest that I've had for a very long time. And in particular, when I started out in looking at genomes and DNA, I did it from an evolutionary perspective. The idea that you can compare proteins and compare DNA from different organisms, and through the bits and pieces that are the same and the bits and pieces that are different, you can see what kinds of environmental pressures there have been on organisms over time. And as a computer scientist, it's very interesting not only to find those patterns, the things that are conserved across organisms, but also to try to figure out, well, what is the chain of events that links different organisms together. So what I'm going to try to do in just under an hour this evening is first talk at a more general level about what is DNA and what is a genome and what does it mean to try to find a gene in a genome, so that hopefully you walk out of here with a sense of what somebody who comes at DNA and biology from the computer science world is trying to do. And then for the second part of my talk, I'm going to do kind of a whirlwind tour of the organisms that we are looking at the Scripps Genome Center and in the Marine Biology Research Division. And throughout that, I'm going to drive it with a question, why is marine genomics important? And try to answer that in several different ways. So the first question is, what is a genome? And this is one of my favorite pictures for illustrating genomes because it shows that in the cell we have a bunch of different chromosomes, and each chromosome is highly wound up. All of this is little bits and pieces of DNA. And if we unravel it, it forms a helix. And if we look more deeply, we find that we have base pairs. A's and T's are always joined with each other, and C's and G's are joined with each other, so that DNA winds up looking a little bit like a ladder. And we can read the DNA. So this is where the Russian came in, Nigella. We can actually build translators, parsers, that can read these letters and figure out what proteins are going to be made by a particular piece of DNA. So if we look at the human genome, we have 46 chromosomes in every one of our cells. If we were to unwind that whole thing, it's about 2 meters long per cell. So that gives you a sense of how much DNA is running around inside our body. 3 billion of these A's, C's, G's, and T's comprise the single read of our genome. And that's really 23 of the chromosomes. So it's really 6 billion letters, but it's really all written twice. And that's a very, very large stack of phone books if you were to ask how many letters are on a given page of a phone book. So that's, and again, that's in every single cell. And an open question is, how many genes are there? How many functional units in a genome? So back in I'm not sure exactly of the year at this point. There's a debate even about that. But in the late 80s, early 90s, the Department of Energy and the NIH funded the sequencing of the human genome. And that led to improvements in genome sequencing technology and machines for exploring DNA and proteins. And that led in turn to a plethora of information. But it's always good to go back and revisit the question, why genomics? Why sequence the entire genome of organisms? And the reason is that embedded in the genome is the full set of instructions that nature needs to make and operate a living organism. DNA has the information in it. And what's kind of cool is that, in fact, the genome of every single individual organism, every one, is a little tiny bit different. So we can look at the DNA of every individual, perhaps not twins, but most individuals, and identify that individual uniquely. And that's true whether you're looking at an E. coli cell or you're looking at you and your sister, as long as your sister is not your twin. And this is what I told you about as my motivation up front. It's this notion that all organisms, from bacteria to humans, share many of the same genes. So whether we're exploring the genome of yeast, mouse, E. coli, Drosophila, or C. elegans, Drosophila is a fruit fly, flies around bananas, and C. elegans is a worm, and all of these are model organisms that biologists have been developing over time for exploring genes and genetics. We can take a gene from E. coli and find pretty much every one of its counterparts in every one of these other organisms. We can explore what it does in each of these organisms and try to figure out, well, what is the protein that that gene is coding for doing? So on the biomedical side, for a variety of diseases, and I've just listed a few here, there are genes that are comparable. They're the counterpart in, say, fly or worm to the human version of that gene. And by either knocking the gene out or modifying it in fruit fly, which is pretty easy to do, there are good techniques for doing that, you can explore what that protein that gene encodes means to the cells in the organism. Is it critical to development, as many of the genes involved in muscular dystrophy are? Or does it go wrong in some repeatable way, as we see with genes that lead to leukemia? So that's the motivation. There's a lot of motivation for looking at genomes, and the genome not only of human, but the genomes of many, many different organisms across life. So how do we interpret a genome? So here's a little bit of a cartoon. We've got a whole bunch of letters here. Now this is not A's, C's, G's, and T's. So I thought, well, let's try to illustrate what we do with a computer. Well, what we do is we set up a rule. For example, AGTG gets replaced with a space. So we can write a little computer program that sweeps through these letters, and every time there's an AGTG, replace it with a space. And so now we get something that looks a little bit like Norwegian or Swedish. And we can add another rule. And the rules are coming from people who are in a laboratory and poking around and trying to figure out in very, very tedious ways that, well, A goes to B and S goes to T. And in fact, every single letter of the alphabet can be shifted left one in this cartoon example. So we do that. So we transform S to T and so on. And we're starting to get something that looks a little bit less like Norwegian and Swedish and a little bit more like Dutch. And now we shift every space left. And sure enough, we can actually read it. So when we set up these kinds of rules, we can read the entire 3 billion bases in the human genome. So I like to think of this as, we're on a mission, a detective mission, and that's to come up with these dictionaries, the rules that let us find words in the DNA. So this is actually my Russian dictionary from when I studied Russian and Spanish and French, and I like languages. I think that's what attracted me to DNA. So what are some of the words in a genome? And this is probably one of the more technical slides in the entire talk. Just so that you feel like you learned a little something, the important parts of a genome that encode proteins are made up of 3 letters at a time. And these are called codons. And a codon is 3 nucleotides, or 3 letters, and they go from AAA all the way through to TTT with every single triple in between. Well, 3 of these in microbial organisms are what are considered a start codon. So you can think of that as something like the capital letter at the beginning of a sentence. So in eukaryotes, ATG is the only start codon. So somewhere along the way, eukaryotic organisms, so multicellular organisms, organisms with nuclei, gave up, allowing TTG and GTG to be the start of a sentence. Stop codons, TAA, TGA, and TAG. So there are 3 more codons there. And all of the remaining triplets, every one of them encodes an amino acid. And I say most everyone because these 3 don't encode amino acids. Well, except in mycoplasmas, TGA encodes the amino acid tryptophan. So there's an exception to everything in biology. So for example, this amino acid leucine, L, is the letter that we can read or translate to whenever we see any one of these 6 triplets. And as you see from this example, every amino acid actually has multiple codons. So there's a degeneracy to this code, this way of reading the genome. And what this means is that if we change one letter, so if there's a mutation that changes a C to a G, for example, some of those mutations will do something like change leucine to valine, because GTC encodes the amino acid valine. But others, for example, this A to G mutation, would simply be a silent mutation, and it would change leucine. So some mutations really do nothing at all. They're just busyness over time. They don't get selected for. Other mutations actually make changes in the proteins. So, well, I lied. I guess this gets a little bit more technical. So what this is showing you is, here's DNA, and we're starting with the very first base. Frame 1 is what it's called. Here we're starting with the second. Here we're starting with the third. And what we're doing is counting off by 3. And so what I highlighted was the stop codons here in yellow. And every single triplet in between, every codon, can actually be substituted with a letter. And in fact, if you look at all of these, every one of them has a star somewhere, which is a designator for a stop codon. So what is a stop? That's like the period at the end of a sentence. So when you see a bit of protein here and then a star, which is a stop, that's not a protein coding region. It has a stop in the middle of it. So this one looks the best. This one also looks like it could be okay. But it turns out that this is the better one because those leucines I told you about, well, some of those 6 codons for leucine are used very infrequently. They're only used 4% of the time. Others are used 50% of the time. And this is the amino acid sequence that has dominant 50% used leucines. This one has the minority leucines. So that's how we can go about taking DNA, translating it, and then doing these counts. And how do we do those counts? By looking at lots of proteins from lots of organisms. And codons are only the protein coding parts. There are other signals in the DNA. For example, a GT and then a whole bunch of letters followed by an AG is the hallmark for what's called an intron. And I'll let you go home and look that one up. A TAT is the minimal signal for an enzyme to come along and attach a whole bunch of A's right there when that bit of the genome is being read and turned into a protein. And the start of reading a gene has certain words that are usually there, and then there's a minority word here. And just upstream of that, 20 letters in front of this -1 +1 position is another sort of motif that often occurs. So we can start to look for these little tiny motifs, these little words that are in 3 billion letters. So go try to look at those words in the phone book. You need a computer to do it. And so that's what set the scene for me with my degree in computer science and my earlier degrees in Russian, which I think is what gave me tolerance for DNA, to come in and start writing parsers, computer programs that can read the DNA and look for genes and try to find genes that are similar to each other across different organisms. So now we'll move to the second part of the talk here and ask the question, well, why marine genomics? What is it beyond just curiosity, which is a perfectly valid reason for going and investigating something? But what pragmatic reasons might there be? What advantages are there to studying marine organisms versus fruit fly or worm? Well, first is just the novelty, so the curiosity effect. There are some pretty strange and different organisms out in the ocean. Another is that there are spots in the evolutionary tree, over millions of years, where ocean organisms sit there and we haven't yet found organisms on land. And then we have some very, very interesting host-symbiont relationships that evolve in community relationships. And the reason for that is organisms in the ocean are floating around there in the water. They have easy access to each other. They can make friends and have battles and wage war with pretty much nothing to get in the way except a few H2O molecules and some salt. Where on land, in the soil, a microbe has to crawl pretty far in order to go beat up its host or try to become infectious. So the ocean has a plethora of these host-symbiont relationships, and we can study them through studying the DNA of the organisms. Then there's the discovery side of it. The idea that, well, let's just go out there into the ocean, let's sequence as much DNA as we can, read it, look for genes, find proteins, and try to find new proteins that actually have functions that we already know about. For example, phosphatases, proteins that can take phosphates off of other proteins and help you with detergents and washing your clothes. New proteins with novel functions, so proteins that have functions that we don't know anything about, and novel compounds, so proteins that make new compounds that are small molecules. And I'll talk a little bit about this. And then the third area for why marine genomics is we can try to look at the diversity of proteins and come up with ideas about how to engineer new ones. And I'll talk a little bit about using DNA as to develop markers to measure the health of an ecosystem. And I'll also talk about novel model organisms that come from marine environments for studying specific systems that are very relevant to human health. So first we'll take a look at this first box. All right, so let's look at a specific genome. So here's a cartoon and here's a picture of the genome, a genome from the sea, that's called Ostriococcus tori. So this is a little one-celled algae that was discovered in 1999 off Scripps Pier by Brian Palnick and his colleagues. And this Ostriococcus tori organism is what's called a pico-eukaryote. So it's one-celled, it's an algae, it has photosynthesis. And what's cool about it is that it's the smallest free-living eukaryote that anybody has found so far. And Brian found it. And once he found it here, people started looking. So once you know what to look for, it's easier to go find it. And these little stars demonstrate all the places where people have found different strains of Ostriococcus. So it turns out this organism that nobody had ever noticed before 1999, when Brian decided to go and sample ocean water off the pier, is actually everywhere where people have looked for it. And if we look at it phylogenetically, this is an early organism, an early branching organism that, if we continue down one branch of this cartoon of a phylogenetic tree, we have Chlamydomonas, which is a common organism for studying plants, and then we have the higher plants. So the idea is that by looking at Ostriococcus and trying to find its genes and understand how it goes about photosynthesizing and living, we can gain insight into how plants do their thing and how Chlamydomonas does. So when I talk about this smallest organism, this gives you the relative scale. So here we have a bacterial cell, which is very similar in size to the Ostriococcus cell. I'm looking to see, I think I have the size of it actually in white. I'm sorry about that. I can't read it either. And this is a plant cell. And you can see how many Ostriococcus cells we can line up here. Here's a chloroplast. So 4 of these Ostriococcus cells fit in a single plant chloroplast. So this is pretty remarkable. We've got a teeny tiny reduced organism that is totally free living, and it does everything that a plant does in terms of photosynthetic functions. So how big is its genome? Well, it's 16 million of these letters. You'll recall I told you that the human is 3 billion letters. And just to give you a sense in between, E. coli is 4 million letters. Arabidopsis thaliana, or mustard weed, which is a common model organism for studying plants, is 150 million. Sea squirts, which I'm going to talk about a little bit, are 150 million. Sea urchins are 800 million, and that'll be relevant toward the end of my talk. And I put a couple pictures of starfish up here because I like starfish. So what do I do when Brian Palnick comes into my lab and says, Terry, I'm getting a 16 million base genome. Can you help me read it? Well, what I do is turn to my students and my postdocs and say, well, let's load this genome into our visualization tools. So something I've been doing over the years is building computer software tools that will read the ACs and Gs and Ts and actually do things like make pictures with little tick marks wherever there's a stop codon, and boxes wherever there's a series of letters that can be translated into amino acids, and thus is probably a protein. And also I do profiling, so this tells you the percent of A's and G's all along the genome, and it's pretty evenly distributed here for this 48,000 letters, 48,000 bases of Ostriococcus. And something that's really, really interesting to look at, especially in these ocean organisms, is GC content. So recall, G's and C's bind to each other, and A's and T's bind to each other. So we can look at the percent GC of a genome, and one genome versus another genome actually has a different GC content. Every organism, if you take it out to enough decimal places, has a different GC content. It's a little bit like your weight. No two people are ever going to weigh exactly the same if you have a scale that can measure it to a good resolution. So this is very, very typical of Ostriococcus, and I'm going to tell you a little bit about this protein here. But before I do, I'm going to mention that within Ostriococcus, and this is something that we're seeing in ocean organism after ocean organism, we wind up with these lower GC regions and higher GC regions in the same genome, meaning that this organism, Ostriococcus, has probably gained DNA from some other organism somewhere along the way. And this is something that, for a long, long time, people who try to reconstruct phylogenetic trees for organisms really resisted. They did not want to hear about genes being transferred from one organism to the other. And the reason is they really wanted to be able to reconstruct ancestral relationships across organisms. But these shifting GC content regions make that hard to do. And in fact, Ford Doolittle is kind of the leader of this notion of horizontal transfer. And he's taken it so far as to say, well, when you look at plants, they're photosynthetic bacteria, and there was a crossover at some point along the way where DNA from photosynthetic bacteria made it into plants, and that's what populates the chloroplasts in a plant cell, the plant genome. Likewise, the alpha proteobacteria sector of the phylogenetic tree donated the DNA that's given us our mitochondria in our eukaryotic cells. And another area that's kind of interesting of this tree, let me just bring this up. So Rickettsia is an organism down in the mitochondrial area, and bacterial photosynthesis gave us chloroplasts and plastids. This third branch of life, archaea, or archaeozoa, is an area where the information processing proteins, so the proteins that actually read DNA, are shared between eukaryotes, and humans are kind of up here. Eukaryotes and these one-celled archaea. So there's a lot to be learned, not only by looking for ancestors, pure ancestors, but by also looking for genes in an organism that have been horizontally transferred from one organism to the next. So one gene in particular that I'll tell you a little about motivates part 2 of the why Marine Genomics slide. So there's a gene called Polyketide Synthase, so it encodes a protein, or PKS. And this is a modular protein. So it's a protein that's made of building blocks. And it was actually originally constructed, if you will, in bacteria. And each module of this protein, the protein's like a factory, it executes what's called an aldol reaction. So it takes a little compound and adds a little bit more onto it, and this compound just makes its way down the protein. So we can consider these drug-like compounds. So a ketone is the starting point, and then a series of aldol reactions creates this chain. And these compounds are used by bacteria against each other in antibiotic defense. So you can think of these as the missiles of E. coli. And as eukaryotes have gained genes from the bacteria to do this polyketide synthase activity, they're able to make toxins. And heaven knows what they actually might do with cell physiology in a normal cell for pure non-warfare-like functions. And as we look in open ocean DNA, we're finding more and more and more examples of variations of these kinds of genes. So, and I'll just give you a picture. So when I take a gene, so here's that gene that I showed you earlier, and I compare it to other proteins that are also PKS proteins, what I find is the regions of my protein from Ostriococcus that are conserved, that are very close to other organisms. And so what this is showing you is 4 different regions that are close, bounded by regions in between that have been allowed to evolve and change. And in fact, there's more to the story. It turns out that one of these modules is high GC, and it came from a different organism, and these 3 modules came from yet another organism in a different place. So in this single gene, there's an awful lot of horizontal transfer and evolutionary change that's gone on. So that example illustrates for you this notion that we can go out into the ocean, sequence DNA, and find new proteins with known functions or novel functions, and proteins that make these novel compounds with anti-cancer, antibiotic, or immunotherapy type activities. So this weekend, I came back from Norway. I just spent about a week and a half there. And the Norwegians are really hot on using marine organisms to develop new sorts of compounds. And this was one of the ones that caught my attention because I have a friend with knee problems. But there's a company in Norway called Navamedic that just got FDA approval for what's called glucomed, which basically they make out of shrimp shells. And it's a collagen regeneration facilitating compound that's good for osteoarthritis. So I thought, all right, that's kind of cool, because usually when people think about compounds from the sea, they think anti-cancer, antibiotic, but in fact, we can mine an awful lot more. So the Norwegians like to call this business bioprospecting, which I think is kind of a cool way to think about it. So the next thing I'd like to talk about, just a couple of pictures here, is how we might get hints and ideas about how to engineer proteins. So recall that Ostriococcus seems to be everywhere. Well, about 2 years ago, this fellow here, Craig Venter, decided to take his boat, the Sorcerer II, out and go into the Sargasso Sea, which is from this little region right here, here's the United States, and just collect DNA from the ocean. And he was the one who sequenced one of the 2 copies of the human genome, and so he put all of his sequencing capabilities to bear on the DNA that he collected from that part of the ocean. So his DNA, so here are some collectors. So they started sequencing, and they published a paper and made that sequence available, and I'll show you what we've done with it in a moment. But right now they're in the middle of a journey going from Halifax, and they're about down, well, I took this picture of current location right now, so Craig is off Australia somewhere at the moment, collecting DNA and sequencing it. But what's great for me about that is I can make pictures with little dots. No. I can take Ostriococcus and compare the entire genome to this environmental DNA, this ocean DNA that Craig Venter is dumping into the public databases at astounding rates. And in the greater than 90% identity region, what I'm seeing is DNA from other Ostriococcus organisms. So I'm seeing variation in Ostriococcus itself across that map that I just showed you. And when I go down below 90% identity, and it's really, it's a continuum, but 90% identical is kind of a nice threshold for is it close or is it not close. And what's kind of cool is if you look closely, there are no dots in this region here. So there really is a drop-off between things that are very, very close, which is other strains of Ostriococcus, and bits and pieces of DNA that are less closely related. So those represent genes from other organisms. So now if I take that DNA and I translate it to protein and I compare it to Ostriococcus, I can start to see within the Ostriococcus, that little tiny organism that does photosynthesis, what is the variation in its proteins. And when I look further into these other organisms' DNA, I can say, and how have those proteins evolved in the environment over time? So this picture is to shift gears to the very last bit of my talk this evening, and that's to remind you, or to let you know, that even Ostriococcus, the world's smallest free-living organism, has infectious diseases of its own. So these are pictures of an Ostriococcus virus. And that brings us to this part down here. And the question is how to use DNA from the ocean to look for hallmarks of is an ecosystem healthy or not healthy. And I thought this looked like a nice, healthy ecosystem, so I put the picture up here. So Nancy Knowlton and Farouk Azam, two colleagues of mine, have been looking at corals and sampling from the surface of corals. So this is not open water sequencing, but rather coral surface sequencing. And what they're finding is that there are hundreds, possibly on the order of 1,000, bacterial species that are associated with healthy corals. And nobody really knows what they're doing, but they're definitely there. And Farouk drew this cartoon to illustrate kind of the way that all these microorganisms associate with each other. So they gather together and they live on what's called marine snow, which is basically dead cells falling down in the water. They come and they hang out with the phytoplankton, so the little microorganisms are over here. They're just swimming around in the water looking for, something bad to do. And then we have viruses, we have ciliate organisms. So there's a lot going on in this drop of seawater that's captured in Farouk's cartoon. So it's heterogeneous and it's clustered. There are these hotspots. So what we can ask, the way we can use genome technologies for examining something like a coral reef microbial community is we can ask questions like, well, the technological question is, can coral reef microbes be surveyed via DNA chips? Why would we want to do that? Well, we'd like to know what organisms are there. We'd like to know what relative numbers are there. And not just the DNA, it'd be great to be able to get at the RNA and ask what genes are being used in what relative numbers in a given community. So I haven't told you about RNA, but. And we have to be able to tolerate the fact that we can't culture these organisms. We may never even be able to sort them out as individuals, but we may only know that DNA from, say, the surface of a coral reef is a collection of anonymous organisms. And they're often dominated by one microorganism or another. Now, it would be great if we can do surveys of healthy coral reefs and then turn around and ask, well, for, say, a bleaching area versus an unbleached area in the same region, can we detect microbial differences, where the differences in the microbes might be due to viruses that are infecting the coral, viruses that are infecting the actual microbes themselves, or it may be due to compounds that are run off from local fields where farmers are farming in ways that are hurting the coral. And can we compare populations from different coral reefs? So looking at unbleached areas in pristine versus touched, so coral reefs that are near farming areas versus coral reefs that are dying, and looking at diseased spots in all three. Because even the most healthy coral reefs have regions that are unhealthy. That's just the way life is. So how can we go about doing that? I thought I'd illustrate for you one of the more powerful technologies that the genomics field has been developing. And that's to be able to take DNA or RNA from two different samples, so for example, a healthy coral reef and an unhealthy coral reef, and label it. So label all the DNA from one sample red, label all the DNA or RNA from the other sample green, and put both of those onto, this is a close-up of a glass slide with pieces of DNA on it. DNA will find itself. If there is a matching piece of DNA, it will go and bind. So what we're seeing here is spots on this glass slide that are red because on that slide, somebody's already put a piece of a gene that the red DNA or RNA found, and other spots are green. And of course, red plus green makes yellow when we're in the light world. So all the yellow spots are spots where both samples have the same gene in equal abundance. And then the darker spots are places where those genes just aren't being detected at high levels. So this is a kind of technology that we can bring to bear to try to discover and then use DNA markers that tell us, that give us a readout about ecosystem health. So the very last thing that I want to talk about, and I think I'm on the verge of running out of time here, is how we can develop novel model organisms from the ocean for studying very specific systems in human that bear on, or in other model organisms that bear on health. So Vic Vacquier, another colleague of mine in the marine biology division, is studying sea urchins. So he's studying sea urchins because he wants to know, how do sea urchin sperm and sea urchin eggs recognize each other, especially in the ocean when these cells that are running around need to discriminate between sea urchin counterparts and other counterparts. So he's looking for the proteins that are involved in fertilization and trying to understand how those proteins are turning an egg just at the moment of fertilization into something that can turn into an embryo, a sea urchin embryo. And the reason studying fertilization in sea urchin is relevant is that, so here's another sketch of a phylogenetic tree. So up here we have human, fish, and mouse. So these are all vertebrate organisms. So it turns out that sea urchins are very early branching, what are called deuterostomes, or basal deuterostomes. In fact, they're echinoderms. And sea urchins and my favorite organism, starfish, are very, very closely related. So you can think of them as pre-vertebrates or almost vertebrate organisms. So we can study a process like fertilization here, where it's easy to get lots and lots of material, cellular material, and not particularly offensive to anybody, and learn things about the human version of the process. And so what Eric Davidson at Caltech has done is taken these early developing sea urchin embryos, and he's, for 50 genes that are involved in the control of early development, early differentiation, and cell proliferation during that early embryonic development, he's asked what happens when he turns that gene off or when he turns that gene on in sea urchin. What happens over the next 24 hours? And by doing that, he's been able to decipher this dependency graph, or this logic diagram. So for example, here, this diagram is telling me that this gene, KROX, is transcribing and then controlling Wnt8, so it's positively controlling that one, and it's positively controlling this HOX11 and 13b complement of Gs down here. So we can try to decipher which proteins are delicately and precisely controlling which genes of other proteins during development. And we can do this in part with the kinds of arrays that I showed you earlier, that glass slide with DNA on it, so we can look to see what gene is on and what gene is off at which moment in time. So the critical question, and this is really the driving question behind any sort of biomedical question that you would ask, is which genes are used in which cells at which moment in time and under what conditions? And I'm going to end with my current favorite organism. We've been studying the genome of the sea squirt, Siona. So sea squirt is actually even closer to humans, and it's about 150 million bases. Recall sea urchin was 800 million, so it's this ridiculously huge genome that's a lot harder to read. Sea squirt has about 16,000 proteins, and it's very easy to do genetics on. And what's cool about sea squirt is it actually, so these little blue, the blue stained protein here, so these look a lot like tadpoles. These are sea squirt larvae. And the blue area here is an early proto-notochord, so it's the precursor to a backbone in these organisms. So eventually these are going to settle down and grow into adult sea squirts here. So at that point, there's not a lot we can learn about the sea squirts with respect to humans. But down here during this early development time, we can. So just to go back to this one more time, why marine genomics? Well, they're novel organisms. They're in strategic evolutionary positions. We can find novel host, symbiont, and community relationships. There are lots of new proteins with new functions to be found and new compounds. We talked about that a little bit. We can look at diversity of DNA and gain insights into protein evolution and how we might engineer proteins to do things in better ways. And we can look for DNA markers for ecosystem health. And organisms like sea squirt, sea urchin, starfish are great model organisms for studying specific human systems. So in my talk, I have to acknowledge I've shown you work that's due to Brian Palnick, a bit by, well, I didn't show you Doug Bartlett's example in the end, but he's sitting here in the audience somewhere. Vic Vacquier works on sea urchin. Farouk and Nancy are working on the coral reef work in microbial communities. That's especially Farouk's area. Bill Gerwick's lab gave us some early tutorials on that PKS gene that makes the antibiotic, that has the antibiotic activity. And Sheila, Trina, Tristan, and Linda are the 4 people who are the very first people in my lab here at Scripps. And Sheila is actually the person who's been analyzing the Osteococcus genome. So I'll just end on this slide, which reprises what Nigella mentioned, and that's that what we're trying to do in the Scripps Genome Center is set up an environment that has the computation, all the computational power necessary, and the people who can actually write new software tools when necessary to carry out comparative genomics, marine genomics, and environmental genomics. So thank you very much for listening. So the answer to your first question, which is, what was the programming language of my first parser? So my first parser was actually written in Prolog. And in fact, I write most of my software now in Perl. That's just my favorite programming language. I'll write something in C if it needs to be fast. And I still write in Prolog when I feel like having a little bit of fun. And for the rest of you, Prolog is a very obscure programming language that involves thinking in ways that are not normal. So your second question is, was RNAi used in Eric Davidson's work? And the answer is yes, but he actually used what are called morpholinos, which are related to RNAi. So again, short oligos that match an mRNA and turn it off. So yep, that's how he did the knockouts. Yeah, so you're asking about the horizontal transfer. And the question is, what happens when you get a piece of DNA that comes in from the outside? So in Osteococcus, there's actually something very interesting that happens. There's this high GC region, and it has a copy of this PKS gene that I told you about. Well, that PKS gene encodes a protein with a certain sequence. And like I showed you, if you can change an A to a G and it won't matter, and in some places, and you can change an A to a G in other places and it will matter, and vice versa. So it turns out that gene, that PKS, that polyketide synthase gene, has been copied 3 more times into the Osteococcus genome, but those particular copies, their DNA has evolved so that it's lower GC. So clearly this gene came in from the outside and then has been adapted into the Osteococcus genome. It's a really interesting process. Organisms are constantly changing and repairing their DNA. You know, and usually it's an experiment and trial and error, but every once in a while, you gain a good antibiotic compound.
So are we humans also absorbing other?
Yeah, well, a really good example is retroviruses. Anybody who's infected with AIDS, for example, has the HIV virus goes into the genome and does all sorts of things. Hepatitis virus, I think, is the same way, but HIV is a really good example of a retrovirus, an RNA virus that turns around and integrates into the genome. So the question is, what kind of genetics has gone on with organisms in the ocean? Well, the organism that's the best developed for kind of high-level genetics, what we would think of as eukaryotic genetics, is actually sea squirt. That's been well studied, and people can cultivate it in the lab and have many generations of sea squirts, and now there are sea squirts that you can take one and make many. So that's been developed. Sea urchins, however, we can't actually make them make baby sea urchins in captivity, so we have to keep going out and collecting them. It's hard, actually, to make ocean organisms make new baby ocean organisms in captivity. So that's one of the challenges about marine biology. Yes. That's a really good question. So how do genes get moved from one organism into another? Well, at the bacterial level, there are these little tiny mini genomes called phages that, mini genomes that are in small things that are basically bacterial viruses. So a phage is a bacterial virus. Phage genomes can pick up DNA from the organism that they're infecting, and then when they go onto another organism, they can leave it behind. So at least in the bacterial world, the hypothesis is that phage, or bacterial viruses, are responsible for moving DNA around, which goes a little bit to your question about HIV and human genome and virus DNA moving around. So it's not clear how organisms would acquire DNA in other ways. What people speculate happened with photosynthesis is that a photosynthetic bacteria got absorbed by another organism, and a eukaryotic organism, and when it did, that became a proto-algae. But all of this is trying to figure out, given what we know about biology and the fact that organisms like to eat each other, eating each other incompletely could lead to capturing the DNA of the other organism. They do. So there's introns in Ostriococcus, little tiny ones, and there's intergenic DNA. Although what we're finding more and more is, at least in the human genome, there are these repeat regions, but those are responsible for bits of the genome getting longer and getting shorter, and they're kind of hot spots for evolution. And then there are just regions where the DNA is very tightly wound up. So it's not clear that anything is really truly junk DNA, because even if it doesn't encode a protein, the more we learn about DNA, the more it seems that these A's and C's and G's and T's are being used for structure, to make parts of the genome accessible, more or less accessible. So how important are mitochondrial genomes? Is that what you're asking? Yes. So the mitochondrial genomes evolve very, very, very quickly. So in fact, you can track populations, for example, a whale population, this is what the fisheries folks are doing here, by looking at very fast changing bits of mitochondrial DNA. And of course, since the mitochondrial DNA is passed down maternally, you can actually trace populations that way as well. So mitochondrial DNA changes quickly from one organism to the next, and the mitochondria is responsible for a lot of the energy processing in the cell. So the mitochondria are critical to eukaryotic cells, and those are a mystery also. Where did the mitochondria really come from? That's not completely clear either, hidden in the depths of early evolution.